that allows researchers to ask sophisticated questions of a lot of the omics data that is being made publicly available. The lead scientist for the SPP is Neil McKenna, who's an associate professor in molecular and cellular biology at Baylor College of Medicine. And presenting today is Scott Oxner, who's currently the biocuration lead for SPP, and before that, the Nuclear Receptor Signaling Atlas. He has a background in reproductive molecular endocrinology and is an expert in biocuration and meta-analysis of transcriptomic datasets. So without further ado, I'll let Scott take over.
All right, thank you for the introduction. So as was mentioned, the title of today's webinar is Signaling Pathways Project, Putting the R in Fair Data. And by R here, we mean reusable, and that's what we're stressing. So much of the archived omics data set are rich sources of information on cellular signaling pathways. And those include expression arrays and RNA-Seq, as well as ChIP-Seq data. Now, the problem with a lot of the archived omics data sets is they're not generally followed up for further study. And typically what you see in a publication are a small number of genes that have the highest or most significant fold changes. And these are the ones selected for more detailed mechanistic analysis and are further published. The problem or the issue here is that the majority of the data on the array or a ChIP-Seq experiment is largely ignored, not followed up on, and tends to get buried in a public repository somewhere. The other issue with archived omics data sets is that they're not really easily accessible or reusable to what I would consider your average bench scientist. For example, at least in our experience, 40 to 50% of the publicly archived data in GEO does not have an associated citation with it. And even though they include contributors, it still makes it very difficult sometimes to find the original data and track down questions that you may need to ask or clarify. The other issue is the actual data itself. It's just a flat file of numbers, and in most cases, a largely uninterpretable ID or reference that your average bench scientist just doesn't have the quick skills in converting into something intelligible. It's akin to, if you consider the research literature, you're really looking at a well-organized, nicely folded closet to something. The omics data archives tend to be something you might find in your teenager's closet, just an absolute mess that makes no sense, and it's extremely difficult to find things and make sense out of any of it. And about 15 years ago, Neil and colleagues started the Nuclear Receptor Signaling Atlas, and I joined their efforts. And one of our main goals was to enhance access of omics, of archived omics data sets in the field of nuclear receptor signaling. And I think we were very successful in these efforts, and we learned a lot of lessons along the way that we were able to apply to our current efforts, which is the Signaling Pathways Project. And really what we've done is expanded our scope from nuclear receptors to basically all signaling nodes that exist. So we're looking at multiple different receptors, multiple enzymes, multiple transcription factors, and conodes. And SPP consists really of two main sections of data. There's the transcriptomic data, which is expression array and RNA-Seq. And the majority of these data sets are derived either from the gene expression omnibus at NCBI or array express at EBI. The other section of data in SPP are systromic data. And these are largely derived from the sequence read archive at NCBI, and we actually use the process data from the ChIP Atlas group in our database. All right, so let's switch to demonstration of what SPP is about. So we have two main sections in SPP. One are the archived data housed in datasets, and then we also have a tool called OMINER, and this is a tool for interrogating the data inside the datasets. So let's take a look and see what datasets actually look like. And remember, I mentioned earlier that there are two main sections of data, transcriptomic and systromic. We also have three easily usable filtering mechanisms here to drill down into a particular data set. And so let's do that. Let's take a look at, for example, receptors, and let's look at catalytic receptors and the insulin receptor subfamily. And let's further narrow that down to metabolic physiological systems, and within those, let's pick specifically liver. So I should say here at the outset that the focus of the rest of the talk using SPP is going to concentrate really on insulin receptor family signaling and cholesterol synthesis, two topics I hope would be very relevant for our NIDDK listeners. I really picked these filters specifically because I wanted to highlight this data set here. If you click on the link, it takes you to the SPP dataset view. So a large part of the metadata that we collect and collate for each dataset is a response to the FAIR standards. We want to make our datasets, findable, interoperable, ideally reusable, and accessible. And to those efforts, each one of our datasets typically does have a name and a description. And for transcriptomic datasets, you can find the node that is being interrogated. Here, it's the insulin receptor. You can also find the species and the general tissue. The description will tell you, for example, here, this is a data set dealing with insulin receptor knockout in wild-type mice and liver isolated from those mice. Each one of our data sets is minted with a DOI, which means it's easily locatable. We also include links to the underlying transcriptomic data, in this case, a geo data set. And further, if someone would wish to cite this particular data set from SPP, we include language for that citation, as well as a couple of tools to easily download that citation into your manager of choice. But equally important here is we also link and provide the citation for the original article that the data was originally generated from. So when you look at a particular dataset view, you see the meta information, and then you also see the experimental information. This particular dataset had a single experiment, insulin receptor knockout versus wild type. And the data is initially presented to you in a scatter plot. And you're looking here at the roughly top 50 up and down genes that were significant to a 0.05 significance value. I'd also like to point out that each one of these data points is interactive. And in the dataset view, you get extra information. For example, you get assembled the fold change that was found and the p-value associated with that fold change. I'd also like to point out, but I'm not going to actually demonstrate at this point, is that each one of these symbols along the left-hand side of the scatter plot is hyperlinked to a OMINER search of the database. So if you found your favorite gene and you wanted to do a quick single gene search that's very easily done from this link here. We'll return to that. So that is a transcriptomic data set. Let's take a look, for example, at a cystromic data set. And again, we're going to use those filters to show an example of, we're going to look at transcription factors, and in this case, let's look at basic helix-loop helix transcription factors, and specifically, I'd like to look at SRE-BP factors. Remember that we are keeping to a theme of the insulin signaling and cholesterol synthesis, and the cystromic data set I'd really like to highlight is this one here, analysis of cystromes in human cell lines, and that's because I want to show you a specific ChIP-Seq experiment where, let's see, here what we're looking at are hep G2 cells that were treated with insulin and then were chipped for SRE-BP. So this is the data set view for cystromics data sets, and now these individual data points are clickable and you get extra information. Here you get the gene symbol. Now what you're looking at, instead of a full change, is the max score. This is the peak score for the ChIP-Seq experiment, as well as a Q-value for its significance. Now I picked this particular chip because I wanted to highlight a specific gene. This one here, a DHCR7, which is the 7-dehydrocholesterol reductase gene. Now this particular gene is a rate-limiting step in cholesterol synthesis. It's absolutely critical for de novo cholesterol synthesis. It converts 7-dehydrocholesterol to cholesterol. So without this gene, you don't have de novo cholesterol synthesis. Last important step in the process. So we'll come back to this gene. And again, just as you saw with transcriptomic data sets, rather, we also employ the FAIR standards for our cystromic data sets. Each one of our cystromic data sets is minted with a DOI. There is a link to the original data in SRA, as well as the ability to cite this particular cystromic data set, and a link to the original article that produced the data in the first place. Okay. So now I would like to demonstrate our search tool that we use to search the actual data set and the data in the database. Now there are 4 mechanisms to search Signaling Pathways Project database. You can use a single gene. You could provide a gene list, or you could use a gene list proxy by using a gene ontology term. And last thing we'll go over is the consensome. Now you can use each one of these search mechanisms for hypothesis generation, which is how we stress our reusability of our data. So let's start out with a simple single gene search, and the gene I mentioned before, DHCR7, that critical gene in cholesterol biosynthesis. You simply type in the symbol and select it, and we're going to leave these filters at their default levels. So we want all signaling pathways, all species, and all physiological systems. The reason for that is you'll tend to return more data as you leave default settings. So the first thing I want to go over a little bit on how to use the regulation reports. So we did a single gene search for DHCR7. These are all of the DHCR7 data points that are differentially regulated, and they're highlighted here. Red is twofold up, at least. Blue is twofold down, and they're all 0.05 as far as significantly regulated. Now, again, these data points are clickable, and you can find extra information within each one of these data points. For example, let's look at this data point. First off, if you're not familiar with what PHA665 is, you're able to click bioactive small molecule button, and that brings up a little information window where you can now link out of SPP to either PubChem or IUPAR, and you can grab more information about what that molecule is. It's actually a MET receptor inhibitor. So what I also want to show is you not only get the gene that's being regulated, it's a single, in our case, it's only one gene because we're doing a single gene search, a fold change, a p-value. You also get the biosample that was used in that study, and then the experiment short name. So in other words, this data point came from an EGF-treated cells versus vehicle-treated cells from a Norway rat. If you want more detailed information about that data set, you click that button, and it brings up the fold change information window. You have the experiment information, the data set information, and again, if I wanted to link back to the SPP data set, I could do that from this point here. So that is the regulation report window in a nutshell. There's different ways to view the regulation report. The default view is the category view. So we're looking at different receptors. We're looking at different enzymes that affect that molecule. We're looking at different transcription factors. And also, we have in Signaling Pathways Project various different animal models and cellular models. For example, adipogenesis or high-fat diet. And these come in handy when you're trying to do hypothesis generation, which I'll illustrate here in a moment. Okay, so let's go over a few points first. What I would like to point out is that that in the fasting and fed state, which are insulin regulate, which is an insulin regulated process, cholesterol biosynthesis, of course, is higher in the fed state and is reduced in the fasting state. We also know that insulin can directly affect cholesterol biosynthesis because it can increase the messenger RNA, the protein, as well as reduce phosphorylation of HMG-CoA reductase, which is another rate-limiting enzyme in cholesterol biosynthesis. So we have links between cholesterol biosynthesis and fasting, which is an insulin-regulated mechanism, as well as insulin-regulating pathways in cholesterol biosynthesis. And lastly, I'd like to point out, we know that, of course, cholesterol is stored in fat cells, but it can also be synthesized in fat cells, but at a very small percentage. And in mature fat cells, I think it's generally accepted that the majority of cholesterol that gets in and out of the fat cells is through transport and not necessarily de novo synthesis. All right, so let's go back to our single gene search. And what I'd like to highlight or point out is this data point here first. And that is, we see our cholesterol synthesis gene being downregulated in an insulin receptor knockout versus a wild type mouse in the liver. And if we further scroll down to our models, we see this large group of upregulated data points. And we can see that DHCR7 is upregulated, or sorry, is induced rather, when you compare adipocytes to pre-adipocytes in the 3T3L1 differentiation model, or whether you're using primary white adipose tissue, DHCR7 is induced. And lastly, when we look at our fasted versus fed models, we can see DHCR7 is actually downregulated in adipose tissue when you're comparing a fasted rat to a fed rat. So already, just doing a single gene search, we have the components kind of a two-tailed hypothesis, and that is that possibly de novo cholesterol synthesis is a component of the adipogenesis process, and we see that here with the DHCR7 being induced during adipogenesis. And we also can make a hypothesis is that that process might be actually driven by the insulin receptor, linking insulin to cholesterol synthesis again. And most interesting is that if you flip this to look at the cystromics data set, a single gene search of cystromics, and we submit that search. And what I want to highlight is I want to highlight the basic helix loop helix transcription factor SREBP. This is that data set that we looked at earlier, insulin treated, and you see this massive ChIP-Seq peak within the DHCR7 proximal promoter of SREBP binding, and it's well known that SREBP is a mediator of insulin signaling in cells. So now we even have a prediction of a mechanism, and that is that insulin through the insulin receptor and through SREBP is driving de novo cholesterol synthesis potentially through the upregulation of DHCR7. So, you know, this is just one way that you can do hypothesis generation with OMINER. Now, as I mentioned earlier, there are other ways. Let's reset our form. There are other ways to interrogate the database. You can do ontology terms. For example, let's just try. We want to look. Let's look at cholesterol, but we already kind of know the answer. But let's look at 7-dehydrocholesterol reductase activity. That's basically the activity of the gene we were just looking at. And let's look at that in receptors, catalytic receptors, and we'll look at the insulin receptor family. We'll submit that search. And sure enough, what you see is that DHCR7 is positively regulated, at least in these mammary cancer cell lines, by an insulin receptor signaling molecule. But you know, the point of a Go term is to use it as a proxy for many different genes. So to do that, let's look at the cellular response to cholesterol, which has many different genes in that Go term. And let's just look quickly at, for example, nuclear receptor signaling. And so here on the left, these are the genes from the Go term that are regulated by nuclear receptors. If we look at the category view, now we can see that the various different nuclear receptors and their ligands are regulating those genes up and down. And if we further look at biosample, we can get a view of what tissues those being regulated in. So that is another way to interrogate the database with a gene list, is to use a Go term. So now let's reset our form one more time. And I would like to show you what you can do with your own gene list. So what I've put together here is I grabbed a gene list here from this study. It's a meta-analysis of the upregulation of the cholesterol process during NAFLD or alcoholic fatty liver disease. And so I picked a gene list from that paper, and we're going to upload that. It is simply a comma delimited list of official gene symbols. I purposely put a bad gene symbol in there so you could see that the system detects bad symbols and gives you a warning in case you would like to correct it. So let's take our gene list of NAFLD-related genes, and let's look, again, let's look at some nice results. Let's look at nuclear receptors, but let's filter that down to the metabolic physiological systems, and let's look specifically at liver. If we submit that, here are on the left, again, these are genes that were in our gene list, and then you see the experimental data points, and again, these are all hyperlinked to more information either about the data set or the biosmall molecule being used in that particular study. And again, you can look at category value to see which nuclear receptors were regulating that subset of genes, and in what tissues. Here we picked liver, so that's why we're only seeing the two different cell lines and experiments from primary hepatocytes. Okay, so the last way that I want to demonstrate how you can generate hypotheses and search the database is to use something that we call consensomes. And for that purpose, we're going to select, again, our insulin receptor family consensome. And before we get too deep into that, I want to explain what consensome analysis is to begin with. So consensome analysis is the ranking of genomic targets according to the strength of their transcriptional relationship within cellular signaling nodes. In other words, what we're doing. Okay, so what we're doing with consensome analysis is here we see that there were 38 experiments that had something to do with insulin receptor signaling from 6 independent data sets. And what we've asked is, for the consensome, what is the consensus? In other words, what genes are consistently or frequently regulated across those 38 experiments? And we rank those based on that frequency. So here is the consensome report, and let's go through it. So for example, the first gene here, PMAIP1, it had a discovery rate of roughly 95%. So in other words, in 98.5% of those 38 experiments, this gene was differentially regulated. Now, when we look at consensomes, we don't look at full change. We only look at those, we don't look at the magnitude of full change. It could be either up or down, because a lot of the experiments in the database are either gain of function and loss of function. So if we picked one direction over the other, they cancel each other out. So we just look at the absolute magnitude of full change, and that the full change be significant. In other words, less than 0.05. So here, the average full change here is just under 2. It's in the 99th percentile. And we give you a significance value. And this is a measure of whether or not this particular gene could have been differentially regulated in 95% of these 38 experiments simply by random chance. This is an extremely low p-value, or in other words, this gene is not occurring by random chance across these 38 experiments. All right, so the first thing you really want to ask yourself about a consensome analysis is whether or not those top ranked genes in the highest percentiles, you want to make sure that they're actually node regulated. So in our case, since we're looking at an insulin receptor signaling node, we want to make sure that the genes that are in that top list here are definitely insulin regulated. And sure enough, for example, cyclin G2 is an insulin-regulated gene in mammary epithelial cells. AMD1, which is a rate-limiting step in polyamine biosynthesis, is also an insulin-regulated gene in rat hepatoma cells. And also here, ERFFI1, better known as mitogen-induced gene 6, MIG6, or in this case, gene 33, it's also expressed, its expression is induced by insulin. So, you know, we feel confident now with our consensome and that some of these top-ranked genes are definitely insulin-regulated. And that's what you should see in something that's claiming to be a consensus. So now what do you do with the consensome? This is where the hypothesis generation comes in. So I'll do a little cheat here, but one way to look at the consensome is to ask, you know, are there any particular pathways that seem to be overrepresented in our consensome list? So for this example, we're going to use autophagy. Right. And simply by doing a search on gene names with an autophagy, you pull out almost, right, you pull out 10 or so genes here that are all within the 90th percentile of an insulin consensome. Now, you might not be too impressed with the fold changes, but remember that these fold changes include ones that are all significant. So they might not necessarily reach a twofold level, but they're definitely significantly changing. So it could be subtle changes, but taken together, this is a hypothesis you could make that potentially insulin signaling is affecting, you know, the autophagic pathway. And what we know about that, of course, is that indeed we know insulin receptor signaling does affect autophagy, and what we know is that this is a well-characterized process, and that insulin affects this process and it's been well-characterized at the protein level. So in the presence of high glucose or in a fed state, you're inhibiting AMP kinase, which then allows the mTOR complex to add negative phosphorylations on the components of the autophagy pathway. In cases of low glucose or a fasted state, low nutrient state, now AMP kinase is able to inhibit mTOR, and it can then add activating phosphorylation events on top of ULK1, which then starts to drive autophagy in the fasted or low glucose state. So this is well characterized at the protein level. But what's not known is, does insulin signaling affect any of the autophagic pathway at the transcriptomic level? And this is where consensome analysis can really help you out. And that's because, for example, if you look at the individual components of this autophagic pathway, ATG14, downregulated. When you look at ULK1, again, downregulated. Let's look at a couple more of these. Before I move on, actually, I want to highlight something about consensome analysis, and that is, you know, ULK1 showed up as a highly insulin-ranked gene, and that one of the 90th, up there in the 98th percentile, and that's because the vast majority of the differentially expressed events for ULK1 are all downregulated, even though they don't quite reach twofold, but they're all downregulated by different insulin signaling here. Insulin itself, two different insulin analogs, and IGF-1. But it's the combined evidence across all of these data sets that is ranking this gene high in the insulin consensome. Now you see that with other of the components. Again, you see a similar thing. The vast majority of those data points are not twofold, but they're significantly regulated, and it's the cumulative evidence that tells you that insulin signaling is affecting that gene and potentially autophagy. So here's a hypothesis that we can take is that, you know, there are 33 autophagic pathway genes in the human genome, and that's out of what, 20,000 genes or so. nearly 50%, or 14 of those genes, are actually in the 90th percentile of the human insulin receptor consensome. So the hypothesis we can make here is that insulin not only is acting on the protein phosphorylation regulation of autophagic pathway, it's also acting at the transcriptomic level. So, you know, the symbiosis between literature and omics data sets can drive can drive discovery in cellular signaling and hypothesis generation. But the problem here is that these published data papers and data sets are, you know, there's gaps. They're not always deposited, and there's all those issues we talked about at the beginning of the webinar. But what we hope that Signaling Pathways Project is able to bridge the gap in between these studies and to allow people to easily search, recall, and reuse the data for hypothesis generation. So I'd like to thank, of course, Neil McKenna, who is the lead PI on Signaling Pathways Project. I'd also like to thank Susan Hilzenbeck, who was instrumental in helping us implement the consensome algorithm. I'd also like to thank the Dan L. Duncan Cancer Center Bioinformatics Shared Resource, and of course, NIDDK and the American Thyroid Association. So thanks, and if anyone has any questions, Neil and I can take those now. No one? No. Hello?
I think everyone's muted, so if someone does have a question, you would have to unmute yourself or type the question into the chat.
Okay, I'm not seeing any questions in the chat either.
Nope, either am I.
Okay.
Oh, hold on. Here's one. So this came in from one of the attendees. Is it possible to do these for data that are not in any database? So
absolutely. We're more than willing to collaborate and work with anyone who has ChIP-Seq, RNA-Seq, or transcriptomic data. We can put it into STP, and if it's not published or it needs to be, it can't be released, we can also work with them on on that point if it can't immediately go public, or if maybe someone has a favorite data set that's in Geo that we haven't imported yet, and they would like to tell us about it, by all means. We're all about getting as much data into Signaling Pathways Project as possible, because that is what's going to improve the reusability mechanism. Just contact Neil McKenna or myself, and we can go from there.
Okay. Any other questions? So another question has come in. Is it possible? Ooh, hold on. So my chat's.
So,
let me repeat.
Is it possible to combine with proteomics and metabolomics? In theory, yeah, it's possible, but right now some of the issues with that are standardizing metabolic names and symbols, and it makes it very difficult to cross from one publication to another, just to make sure you're dealing with the same metabolite. Right now, we are funded to work with ChIP-Seq and transcriptomics data. So we, of course, would be more than willing to talk with anyone who is interested in incorporating metabolomics and proteomics data.
And then there was another question that came in. Have you ever tried to integrate multi-omics platforms, including chromatin remodeling, ATAC-Seq, in your model of signaling pathways?
Oh, we haven't attempted that at the moment. But, you know, anything like that is possible. But we haven't had our hands on any that type of data at the moment.
Okay. Last call for questions. Okay. Thank you, Scott, for a very informative webinar. And a question just came in about the slides. The webinar, we've been making the webinar available, but we could also potentially make the slides available. And so we can make those, if you're fine with sharing the slides, Scott, we can make them available via.
I will.
Okay.
I'll send the slides. I'll send the slides so you guys can post.
Okay, and then we'll post them through our dkNET. I guess it would probably be SlideShare, but I'll check with Koei on that.
All right. Yeah, I'll email her. We'll work it out.
Okay.
All right. Thank you, Jeff.
And thank everyone for attending. And then the next webinar on May 10th will be an overview of the final dkNET 3.0 release. So give an overview of all the dkNET tools in the new release. And so hope to see people there.